In this video, we will discuss SNPs, which is short for single nucleotide polymorphism. By the end of this presentation, you will know 1. What an SNP is, 2. The first major use of SNP-related evidence, 3. Steps of the SNP testing process and what makes an individual SNP match, and 4. When and why SNPs are used today for DNA testing.
A single nucleotide polymorphism is a DNA sequence that occurs at a single nucleotide. DNA sequences are composed of a chain of 4 nucleotide bases: adenine A, cytosine C, guanine G, and thymine T. So if a certain position in the human DNA sequence is particularly unique, the DNA nucleotide base can be compared and used to make a match.
SNPs were readily accepted in identifying victims of the 9/11 attacks. Since the temperatures that occurred during the attacks so heavily degraded the DNA, SNP testing was used to identify individuals opposed to other methods that would prove ineffective. In 1997, biotech companies took the initiative to isolate 60,000 or more unique positions found in human DNA chains to develop a whole genome SNP-based map. And then in 2003, members of the European forensics DNA typing community developed a project to aid forensic DNA analysis and actually use these SNPs to aid in forensic investigation and DNA matching.
The most common process of testing SNPs is called Premier extension. To match two samples using Premier extension, scientists must, first, hybridize the bases that come before the desired nucleotide, or the SNP. This means that a sequencing reaction will occur as an added synthetic complementary molecule, or the primer, bonds to bases leading up to the variation base, or the SNP. Second, scientists will see if the added synthetic molecule will bond to the SNP. If it is a mismatch, and if the synthetic complementary DNA molecule does not match the SNP, the sequencing will stop. If the synthetic DNA matches and binds with the SNP, it will continue downstream to other bases. Lastly, scientists can do this process with multiple samples, and if two have the same reaction to the same base, that can indicate a match of the samples. This similar reaction is necessary in making an individual match in the context of an investigation as well.
SNP testing is used today for many reasons, one of which being how convenient and cost-effective this method is compared to other DNA sequencing methods. Other methods can be very expensive and time-consuming, however, scientists have created SNP detection methods that are productive in matching DNA. These methods are less expensive and time-consuming and also can still be performed using small or degraded samples of DNA. This is why this method is still being used today in instances where a DNA sample must be matched to aid in an investigation.